One of the experiments in this week's lab is the Kirby-Bauer antibiotic sensitivity test. So it's a disk diffusion test, and we use a specialized media called Mueller-Hinton agar. Now, Mueller-Hinton agar has a pH of 7.4. It's poured 4 millimeters thick, and it has a pH somewhere between 7.2 and 7.4. So the discs then, antibiotic discs, are put on top of a plate that's been spread with bacteria, and the antibiotic will actually diffuse out. And after you incubate, you will then measure what are known as zones of inhibition. A zone of inhibition is a clear area around the disc where no microbe is growing. So here's some examples then. So you can see this clear area right inside here is the disc itself. Then you measure the diameter from one side to the other, and you compare that number then to a chart. And the chart will tell you whether or not the bacteria is going to be susceptible to the antibiotic, resistant to it, or have what's known as an intermediate level of sensitivity. So if it is susceptible, then that means you can use it. So that's something you want to keep in mind. Susceptible, you use it. So this is good type of testing because if you have a patient with an infection, you want to make sure that you can treat them with an antibiotic. So always use a susceptible one. However, sometimes it turns out that the bacteria is going to be resistant to it. So when you measure that zone, it will fall within the resistant range, or it'll be that number or lower. Now, intermediates give a borderline response. And what I always tell my students is that if you measure intermediate, there's a good possibility that your bacteria is becoming resistant to that antibiotic. So only use intermediate as a last resort. So again, you should know for your exam that this is a highly standardized test. So the media itself, Mueller-Hinton agar, pH to 7.2 to 4 millimeters thick. The antibiotic concentration is also important because we want to use an antibiotic that's physiological relevant. So what that means then is that you don't want such a high concentration that it would kill you as well as the bacteria. So we do standardize for that. Also bacteria concentration. And one thing to keep in mind with bacteria concentration is the types of bacteria we normally use in the lab for this test would be E. coli and Staph aureus. They can replicate every 20 to 30 minutes. So you want to make sure you get your discs on within that period of time before the bacteria have a chance to actually replicate, because the more bacteria that are there after 30 minutes, the smaller those zones of inhibition will be, so you'll get a false reading. Now, antibiotics work in different ways, and they can target your cell wall, cell membranes, different cellular processes, and proteins. So we have a couple of different ones that we use in the lab, so please know these. First one is ciprofloxacin. Ciprofloxacin will inhibit DNA gyrase. Polymyxin is another one. That will damage cell membranes. We have antibiotics that will actually inhibit the cell wall. A good one for that is penicillin. And what it specifically does is it inhibits transpeptidase. Inhibitors of essential metabolites would be things like trimethoprim, which will inhibit folic acid. Then we have some that will inhibit protein synthesis. Those three are streptomycin. Streptomycin binds the 30S ribosome, which causes messenger RNA code to be read incorrectly. Tetracycline, that will bind the 30S ribosome as well, but it inhibits transfer RNA from binding to the A site of the ribosome. And lastly, chloramphenicol. That binds the 50S ribosome and inhibits peptide bond formation. Please know all of these for your exam and for your quiz. Now, again, when you measure your zone of inhibition, you're going to measure the diameter, okay? And then you compare to a standardized chart that can be found on page 41 of the lab manual, and I'll pull that up in a sec. So get your ruler out, you measure from here. There's a really good little virtual lab that I uploaded for you guys. Just click on the link and you can practice. It's got the zones of inhibition chart for you. You measure it with the little ruler. Click on whether you think it's intermediate, whether you think it's resistant or susceptible, and it'll tell you if you're right or wrong. So in your lab manual, guys, then, on page 41, this is the chart I was talking about. So again, if your number that you measure is the same as this column here, the resistant column, so the same or less, the bacteria is resistant to that antibiotic. If it falls within the range, intermediate range here, and each one, if you take a look, has a different range, that means that the antibiotic has some effect, but that the bacteria may become resistant to it. So only use that as a last resort. And if you measure in the susceptible range, this number or larger, that means that your bacteria is susceptible to that antibiotic, so you may use it to treat your patient.